Good afternoon, everybody. My name's Sheila Graham and I'm Chair of the Biochemical Society. And the Biochemical Society and Port Press are pleased to welcome you to this, our second webinar in our new Biochemistry Focus webinar series. So the topics in the series will include different research areas in the molecular biosciences, as well as practical sessions that are intended to support career development. Each webinar will give you the opportunity to ask questions via text, and we will welcome suggestions for future topics and speakers to feature in this webinar series. And please see our website for more details. So today, our invited speaker is Dr. Andrew Hammond from Imperial College London, one of the 2020 Biochemical Society's Early Career Research Award recipients. These Biochemical Society Awards recognise the impact of research carried out in the molecular biosciences by early career scientists. In other words, those people who have had no more than 6 years postdoctoral research experience. It's expected that the successful candidates will have produced international quality research outputs and be able to demonstrate ambitions and aspirations consistent with the potential to achieve world-leading status. Research areas include, but are not limited to, genes, energy and metabolism, signaling, biotechnology, biological systems, molecular structure and function, and cells. So Andrew, or Drew as he's known, completed his PhD at Imperial College London working on gene drives. Gene drives have incredible potential to modify entire populations of malaria mosquito by biasing their own inheritance. Drew's PhD was focused on finding suitable targets in the mosquito genome and establishing CRISPR technologies in the mosquito to test its potential as a gene drive. His primary PhD research demonstrated the first gene drive system designed to suppress populations of the malaria mosquito, including the first use of CRISPR in the African malarial mosquito. Drew has been awarded a Sir Henry Wellcome postdoctoral fellowship to initiate a new line of research aimed at understanding the neurobiology underlying mosquito attraction to humans. This work is being undertaken at Johns Hopkins University in the States, Imperial College London, and the University of Oxford. Today, he will be presenting his award lecture on building gene drives to beat malaria, where we will get to know more about his work and how it has been developed to focus on targeting sites in the genome and the first CRISPR-based system to bias the sex ratio, key process needed to develop several strategies for pest and vector control. At the end of the seminar, there will be questions, and you as attendees can send in your questions during the talk or at the end of the talk. So now it's time to hand over to Drew to give his presentation. Thank you, Drew.
Thank you very much. Let me just share my screen. Okay, perfect, brilliant. So I'm very, very happy to be talking to everyone today. It's a real honor to have been awarded the Biochemical Society Early Career Researcher Award. And I have to say that the Biochemical Society has actually supported me not just in this, but they've helped me to attend conferences and in other parts of my career as well. So I am very thankful, and I really hope that I can give you guys a nice talk about the work that I've been doing for the last decade on the development of gene drives. So it might help to explain first why it is that I work on the mosquito. And actually, it's because the mosquito is the world's deadliest animal on Earth. So through the transmission of diseases like Zika, dengue fever, and yellow fever, it causes the death of around 750,000 people each year. But the vast majority of these deaths are caused by just one disease, and that's malaria. On its own, it is responsible for around 430,000 deaths every single year. And we find the disease throughout the tropics, because this is where you find the malaria mosquito. But the disease is not equally spread across the tropics. You find the worst forms of it in sub-Saharan Africa and in India. And part of the reason is not just because of the mosquitoes that are there, but also because of the parasites. And so you have different forms of the malaria parasite, some of which are more deadly than others. And these are most commonly found in sub-Saharan Africa. You also have the most efficient vectors of malaria in sub-Saharan Africa. So together, that accounts for the reason why sub-Saharan Africa has more than 90% of the deaths from malaria. So you might ask, how do we currently fight this disease? And the most obvious thing to do is to go after the parasite. And indeed, we have technologies to do this. We have anti-malarial drugs, and we have the development of some new vaccine. But unfortunately, in both of these cases, it's not something that's likely to lead to eradication of malaria. When you think of a vaccine, you often think of something that's 100% effective and very long lasting. But right now, the vaccine that we have provides a very low protection, less than 50%, and it doesn't protect some of the youngest people who are most at risk of serious complications from malaria. And with anti-malarial drugs, it's true that we can use these and they are excellent as a preventative tool, but unfortunately, long-term use of the drugs is not the best thing for your human health, and it leads to selection for parasites which are resistant to these drugs. So historically, we've actually relied on targeting mosquito vector as an effective way to control malaria. And we have gone from more than 1 million deaths in the year 2000 to less than 500,000 now. And it is almost entirely because of the use of indoor residual spraying and insecticide-treated bed nets. And these strategies are the use of insecticides to kill mosquitoes before they have bitten someone and before they're able to transmit disease. But it is also effective if a mosquito were to bite someone and pick up the parasite, they will rest on the walls of a house. And by spraying insecticides on that wall of the house, you're able to kill that insect before it can transmit again to another person. But when we talk about the mosquito, we need to be careful, because there are more than 3,500 mosquitoes worldwide, and very few are actually capable of transmitting disease to humans. We know that there are 3 species that transmit more than 95% of malaria in Africa. These are Anopheles gambia, Anopheles scolitii, and Anopheles arabiensis. And we're now seeing significant transmission from another group of Anopheles vectors called Anopheles funestus. But the vast majority of mosquitoes are not transmitting disease to humans. And even when we talk about these species, not all of them transmit disease. It's only the female mosquitoes. And that's because the female must bite the human to take blood so that she can produce eggs. So the males do not bite in the lab. Perfect. So apologies, everyone, but I think we can get very quickly back to the point. So the idea is that what we want to do is to modify mosquitoes, and we want to do that in a way that's useful for vector control. But if you were to modify some mosquitoes in the lab, how do you get them into Africa? How do you genetically engineer mosquitoes on an entire subcontinent? So for that, we're going to introduce gene drive. And what I want to do is introduce a theme. So in this slide and the remaining slides, if I show a blue mosquito, this means a wild mosquito, something which is unmodified. And if I show a red mosquito, this is one that's been modified, either with a gene drive or just another genetic modification. So imagine that we introduce any genetic modification into the mosquito, something that we think is going to help eliminate malaria. If this were to mate with a mosquito in the wild, you would only see 50% transmission of your modification, but the other half of the progeny would remain wild. And every generation that goes on, you'll have the same thing happening. And so a modification that you can release 100 genetically modified mosquitoes today, and in several years' time, you will only have the same 100 genetically modified mosquitoes, more or less, because the frequency of that modification will tend to stay the same over time. But if it has a cost, that modification will get lost, and you'll have no impact on the wild population in Africa. So what gene drive offers is a way to spread genetic modifications in a way that's sustainable. So with a gene drive, a genetic modification is not inherited at 50%, but it's inherited in theory by 100% of the offspring. And this means that every time they mate with a wild mosquito, the frequency doubles. And they will then mate with more wild mosquitoes, and the frequency will double again and double again. And so in 20 to 30 generations, you can go from a few genetically engineered insects to an entire population that contain this one modification that you release. And we can use it to spread anything we want in theory. So we could spread mutations that make your mosquito unable to transmit malaria, or perhaps it makes the mosquitoes so they don't want to bite humans anymore. But the most interesting thing, and perhaps the most important for malaria control, is to find a way to spread a modification which kills and destroys the most dangerous mosquitoes without affecting those that cannot transmit disease. Now, this is going to seem like a paradox, because the way gene drives work is they work through reproduction. Every time the mosquito produces offspring, the number of gene drive progeny is double what you'd expect by Mendelian inheritance. And yet, I'm going to tell you about how we can spread a gene which makes mosquitoes sterile. So how can you spread a gene if they're unable to reproduce in the first place? So it relies on two rules. The first rule is that it needs to make your mosquitoes infertile, but only if they're female. So it's not going to affect males. And the second rule is that it will only make them infertile if they inherit it from both parents. So if one parent is wild, they'll be unaffected, but if both parents are modified, it will be a sterile female. And males are always unaffected, and females are fine as long as one of their parents was wild. So let's see what happens. You have your gene drive mosquito in red, and it mates with the wild one, and they will produce only gene drive offspring. And those offspring will go on to mate with more wild mosquitoes, and they'll produce only gene drive offspring. And so it starts to spread through the population. But now you're going to have a situation where you have many of these gene drive mosquitoes, so they're going to start mating to each other. And when they mate to each other, now you have female offspring which are going to be sterile because both their parents contain the gene drive. But the gene drive is still able to spread because the males and the heterozygous females are completely fertile. So this allows us to achieve population suppression. The idea is that you would release potentially a single release of a low number of mosquitoes just once into a population in Africa. And the gene drive initially is very rare. So it's going to keep mating to wild mosquitoes, and it's not going to find other gene drive mosquitoes to mate to. And that means that it will just spread and spread and spread. But as it spreads, eventually they'll start mating to each other, and they'll start producing sterile females. And at this point, the population will start to go down. And so from a single release, you can modify the entire population, but also suppress it. And if you reach a low enough number of mosquitoes, you could eliminate the whole of malaria, at least at a local level. So it's a very exciting technology. So you may be thinking, how quickly would this happen? So earlier I said that mosquitoes reproduce, that the gene drive can spread to fixation in 20 to 30 generations. Now a mosquito reproduces every 3 weeks. So 20 to 30 generations is just a couple of years. Now, of course, that's the very optimistic scenario, but it is theoretically possible that a single release of mosquitoes in a small population could reach fixation in just 2 or 3 years. So how do we build them? Well, the idea came about in 2003 from Professor Austin Burt at Imperial College London. And back then we didn't have CRISPR, but the idea relied upon any sort of endonuclease that we could re-engineer to cut DNA. But in 2012, CRISPR technology allowed us to make this a reality. And so I'm going to explain the technology as we do it now. So imagine we have 2 chromosomes of the mosquito. We've got one on the left and one on the right. And on the chromosome on the right, we're going to insert the genes for CRISPR. So this chromosome has been edited to contain this red region, which is the Cas9 gene and the guide RNA gene. And we're going to program the guide RNA to recognize the homologous chromosome. So these chromosomes are identical other than the fact that one contains Cas9 and guide RNA and the other one does not. But we're programming that guide RNA to recognize a gene on the homologous chromosome. In fact, it recognizes its own sequence. So when you express the guide RNA, it's going to come along and it's going to cut the homologous chromosome in this yellow sequence. But now that that chromosome is broken, it needs to repair. And the best way to repair a broken chromosome is using your homolog as a template. But if it does this, it's going to copy the instructions for Cas9 and guide RNA inside. So what was previously a heterozygous modification is now converted into a homozygous modification. And we call this process homing. And that modification, the Cas9 in the guide RNA, we call that the gene drive. So I want to give you an overview of the strategy and how it works now. Initially, we create a modification in the mosquito, and that modification is going to express Cas9 and guide RNA in the germline of the mosquito, so that in a heterozygote, that modification will cut the homologous chromosome, and it's going to copy inside by homology-directed repair. Now, because the gene that we're going to cut is required for female reproduction, when the mosquitoes are homozygous for this, they will be sterile, but when they're heterozygous for this, they will be fertile. So the gene drive spreads through the population, and on the right, you see heterozygotes being produced, and they're fertile and continue spreading. But on the left, you see the production of homozygotes, and the females will be sterile, and the males will be completely fine, and continue spreading that element. So that over time, you'll reach a population suppression from essentially one release of these mosquitoes. So the next question is, how do we build them in a lab? So when I started off in my PhD, we didn't have CRISPR technology, but it had been published in Cells. And we got very excited and we said, okay, what if we could program CRISPR to recognize mosquito genes? And as everyone knows, CRISPR works very well across a broad range of organisms. So we used CRISPR to destroy genes that we thought mosquitoes might need for female reproduction. And we destroyed genes that they would need, hopefully, to produce eggs. So we inserted in a GFP modification that allowed us to track these mosquitoes. And we saw that in the heterozygotes, they were completely fertile. So in this graph, you see the number, all of these dots are the number of larvae that a female is able to produce. So the wild type mosquitoes produce about 80 larvae on average. But when we disrupted each of these genes with the code names RGUP 011377, RGUP 005958, and then the last one 7280, when we disrupt these three genes, they were completely fine when we break one copy. But when we broke two copies of those genes, the mosquitoes were completely sterile in the females. The males were unaffected. So this is exactly what we needed for our gene drive. So the next thing is, what about if instead of a GFP, we place in a Cas9 and guide RNA and convert it into a gene drive? So we used a process called recombinase mediated cassette exchange to swap that GFP modification for the Cas9 and guide RNA. And therefore you convert it from a simple null mutation into this self-propagating gene drive. So to test it, the first thing we did is we took mosquitoes that contained one copy of the gene drive, we mated them to wild mosquitoes, and we simply counted how many of their offspring are glowing red, how many of them contain the gene drive, and how many of them do not. If it doesn't work, it should be 50%, and that's this dotted red line here. But if it's working, you should see more than 50% of the offspring are red, more than 50% inherit that genetic element. And we did this at each of these three genes, and we tested the ability of this reaction in both males and females. And in every case, we saw extremely high inheritance of our construct, as much as 100% in the majority of mosquitoes we tested, and an average of 99.6 for one of the strains. So to test it further, we said, if it's able to bias its own inheritance, it should be able to spread if we were to release these mosquitoes into a population of wild mosquitoes kept in the laboratory. So we did exactly that. We had a group of wild type mosquitoes and we seeded into that. We had a population of around 600 mosquitoes, and we started with half of them were modified with the gene drive and half were not. And then we left them, and we left them generation after generation just to mate and breed and see what happens. And we saw that every generation the gene drive was increasing in frequency, and it went from 50% to 80% in just 7 generations. This was the first demonstration that gene drives can spread in any organism. So we're very excited. But when we followed this cage for longer, what we found was that the mosquitoes were generating resistant mutations. So when the gene drive started spreading initially, it was very exciting, but then the gene drive got lost. It did exactly the opposite of what we wanted. And eventually the mosquitoes had no gene drive at all, but they were completely healthy. So it was a failure in some ways. So we looked at thinking, how does this happen? And it's not something that caught us by surprise. It's actually something that we predicted right from the start, and it was part of Austin Burt's theory in 2003. He predicted that your gene drive is programmed to recognize and cut a very specific piece of DNA. So if there's a change in that sequence, it could block the Cas9 guide RNA from cutting. So we would call that a resistant mutation. Now those resistant mutations can arise in one of three ways. They can be generated by the gene drive itself, and they're generated through the process of end joining. So when CRISPR cuts DNA, we know that 99% of the time it will copy using the homologous chromosome as a template. But sometimes you get end joining mutations, small insertions or deletions that will then make it refractory to being targeted again. The sequence changes so you can no longer cut it. But you don't need to have mutations caused by CRISPR. Mosquitoes may contain mutations themselves naturally. All of us contain natural variation that distinguish one human from another, and mosquitoes contain natural variation. So you might choose a region of the genome that you want to cut, but many mosquitoes might have a different sequence there, and so they are already naturally resistant to the gene drive before you release it. And the final problem is that our gene drive is designed to make mosquitoes infertile. So any mutation that stops the gene drive from spreading is going to come under a massive selection. And any cost imposed by that gene drive is going to result in a stronger selection for resistant mutations. So the first thing we did was we said, are all mutations going to be resistant? And it turns out that that's not true. Not all mutations are resistant. If you change the sequence of an essential gene that's needed to produce eggs, the chances are that gene will no longer work. If you were to delete one or two bases, that would be an out-of-frame mutation, and then those mosquitoes won't be able to produce eggs. The real problem is if you get a change in the sequence that your guide RNA is programmed to cut, but the gene is still functional. So think about a synonymous mutation. You could have a change in the DNA that does not affect the protein itself. The protein sequence is still the same, despite having a change in the DNA. So the mosquito would not really know that it's been modified in any sort of way. However, it's now unable to be cut by the Cas9 guide RNA because the sequence is different. So we call those resistant mutations. Whereas mutations that block cleavage but result in a non-functional protein, those would be non-resistant mutations. So we set about addressing this issue by first saying, what if we can make a strain where the fitness cost is not so severe? So with the initial version of the gene drive that we made, we found that the females that were heterozygous, they're supposed to be 100% fertile, but they weren't. They were giving us 7 larvae on average instead of the 60 or so that we see in wild mosquitoes. So we thought perhaps the Cas9 is being expressed all throughout the body and causing havoc and causing mutations that make the mosquito very unfit. So we looked at better ways to restrict expression of the Cas9 guide RNA to make sure that the mosquitoes are healthy. And to do this, we used different promoter sequences that were very specific to the germline. So when we used these nanos and ZPEG promoters, we saw the fertility increased from the 7 larvae that they were producing on average before to 50 or 60. Now 50 or 60 was equivalent to the wild type. So what we found is just by restricting your CRISPR activity, your mosquitoes became much fitter and the selection pressure for resistance is much less. And they still worked very well. They still worked incredibly well as a gene drive. So with our original gene drive, it spread for the first few generations, and then it got lost. And that's what you see in this panel A. And in panel B below, what we did was we sequenced the genome of the mosquitoes every generation after release. And we looked for mutations, and we looked how is the landscape of mutations changing over time. And you see that initially, many of the mosquitoes contained lots of random mutations, which are these mutations in blue and in red, and in gray is the wild type. But over time, everything turned into these red mutations. And the red mutations are the resistant ones, the ones that are really a problem for gene drive. But when we swapped to this new system, where the mosquitoes are much fitter, the gene drive was able to spread up to almost 100% of the population. So this was incredible. We'd never managed to achieve this before. And the only way we did that was by improving the fitness of these insects so that they were so that resistant mutations had less of a benefit. but eventually, when the population starts to be suppressed, there is still a selection for resistance. So what we saw was that resistant mutations were initially very, very rare, and you see that at the beginning, almost everything is grey. But once the population was nearly 100% gene drive, that means almost all the females are sterile. So the few remaining females that are left that are fertile, they must have had a resistant mutation, and that mutation spread into the entire population afterwards. So everything contained the same 1 or 2 mutations, in contrast to the original gene drive, where you had many, many, many different mutations that were created and selected over time. So what about pre-existing resistance? And this is the thing that I think is most interesting. So we talked before about the fact you can have mutations in your chromosome which prevent the gene drive from working. But not all of those mutations are resistant because they need to encode a functional gene. So we thought, what if we were to find a gene where the nucleotide sequence is the same in every single insect? If it were the same in every single insect, then maybe that implies that there's some constraint to that sequence. Maybe there's a sequence where if you were to introduce even a single base change, those mosquitoes would die or they would be unable to reproduce, in which case they would never be able to get resistant to our guide RNA in Cas9. Now, it seems impossible. And in fact, when we started looking at the genome of mosquitoes, we thought it was impossible. So some of our collaborators, they went to Africa and they sampled more than 1,000 mosquitoes from across different regions of Africa, and they looked, what is the diversity? How often do you find a single base change in the mosquito genome? And it turns out that 1 in every 2 bases is variable. So think about that. We want to find a region that a guide RNA can target. So that's about 20 to 23 bases long. And we need it to be the same in every single mosquito, because if it's not, the guide RNA won't work. And those mosquitoes that we cannot target will be resistant. But what we found in nature is that 1 in every 2.2 bases has a variation. So it seems that there's no chance in finding a sequence long enough that is the same in every mosquito. And that's a real problem for us. So we started looking at what parts of mosquito biology might be more likely to be conserved. What parts of mosquito biology might be the same in every single mosquito and therefore less likely to have this variation. And we looked at the mosquito sex determination pathway. And I'll give you a bit of background on this because it's quite interesting. In all insects, there is an initial signal which tells the insect whether it's going to become a male or a female. And this is determined by the chromosome. But sometimes it might be the presence of a Y chromosome. Sometimes it's the number of X chromosomes. Sometimes it might even be a single gene, like a male determining factor. And it can be different across the insect species. And so you never know exactly what your species might have as its mechanism of determining whether it's going to become a male or a female. A single gene, the presence of a chromosome, or even the number of chromosomes relative to others. But what happens is, whilst the initial signal can be different in different insects, it activates a cascade of genes that ends up with 2 master switches called doublesex and fruitless. And these 2 genes are the same in every single insect. Every insect has these 2 genes, doublesex and fruitless, and those 2 genes alone are enough to determine whether it becomes a male or a female. And this is surprising. You have an initial signal which can be completely different in different animals, and yet they all rely on the same 2 master switches to go and program the development of male and female biology. So we looked at this gene doublesex, and we found that not only is the protein very similar across different insect species, but it has a structure which is the same. It has a part of that gene which is essential to female biology and a part of the gene which is essential to male biology. So there is an exon called exon 5, and this exon 5 is only spliced in the female form, but males do not have this exon. So we thought, this is interesting. Maybe this is a good target for gene drive. Maybe we could destroy exon 5 and we can affect female mosquitoes without affecting the males. So we looked at this sequence, and it turns out that it is one of the most conserved pieces of DNA in the whole of mosquito genomes. And when I say conserved, it's not just the same in every single individual of the Anopheles gambia species, but it's the same in closely related species, such as Anopheles glutzi, Anopheles meris. But in fact, even if you go to the mosquitoes you find in India, Anopheles defensi, you find just one base difference in the region we're targeting. And across the entire region of exon 5, you find just 2 bases different. This is incredible, and it implies that there is some constraint to this sequence beyond just making a protein. If this DNA was important for making a protein and that was it, you should find bases that are different in the wobble position. You should find bases that are different but would encode the same amino acid. The fact that you don't find them in nature implies that this DNA sequence is performing another function. Perhaps it's a binding site for a transcription factor or a splicing regulator. But whatever it is, we know that this DNA seems to be intolerable of variation in nature. So we disrupted this sequence, and we wanted to look what effect does it have when we disrupt this single sequence. Does it kill the mosquito, or does it only affect females? So here you can see a picture of the male mosquito and the female mosquito below. And the males are distinguishable from the females because they have these big bushy antennae, and at the end they have these claspers which are facing down, and they use them to catch females when they're flying to mate with them. When we destroyed this exon 5 by inserting a GFP, the female mosquitoes that were genetically female did not develop normally. They developed into an in-between sex. They developed as these intersex mosquitoes or pseudo-males. They had many features of males, but they were unable to bite, they were unable to reproduce, and they could not mate with males or females. So we converted this into a gene drive. And I want to cast you back to what I mentioned earlier. We made a gene drive in the past, and we targeted a gene that mosquitoes need to produce eggs. And it spread through the population, but very quickly they developed changes at the nucleotide level that made them resistant to the gene drive and resulted in it being lost from the population over time. When we did the same thing in this ultra-conserved region, our gene drive spread from 25% to 100% in 11 generations and resulted in 100% modification. So that's one single release of these gene drive insects were able to spread through an entire population of mosquitoes that we had contained in a cage in the laboratory, and there was no evidence of resistance. But now think, what did that do? When that gene drive spreads, it's going to do something. It's going to destroy that exon 5. And when you destroy exon 5 of this gene doublesex, you make females that are unable to reproduce and they're unable to bite. So if these females are unable to reproduce and bite, then what happened once every single individual was modified? So we counted the number of eggs every single generation. And initially they were producing around 30,000 eggs every generation, and we counted these one by eye. But after 8 to 12 generations, every mosquito was converted into one of these intersexes, and the number of eggs went down to 0. So we were able to completely eliminate mosquito populations by releasing these gene drive insects just once and letting the gene drive spread on its own. And as it spreads, it reduces the number of females that are able to reproduce and replaces them with intersex females that are no longer able to reproduce. So we anticipate that in nature, targeting just one site might not be enough. Even though we haven't found resistance in our laboratory testing, it doesn't mean that it can't happen. So what we've done is we've said, what about if instead of targeting one ultra-conserved site, we were to target 2? Now here what would happen is the gene drive would cut in 2 different places, very close by, and that would result in the gene drive copying just like normal. But if you were to have a resistant mutation at one of these sites, so let's imagine a single change in the DNA that stops your guide RNA from cutting, it would only stop cutting at one site but not the other. So actually, this would not be resistance. So you can actually mitigate against resistance by cutting in several places at the same time. And it doesn't matter which of those sites is able to be cut, the gene drive will copy inside. Now what you might be thinking is, okay, fine, this will delay resistance. You'll start off getting resistance at site A, and then over time, you'll then accumulate the second resistance at site B. But that's not true. Because if you were to have resistance at this site on the right, this R in red, but you're still able to cut to the site on the left. That whole chromosome is going to be cut and replaced with a gene drive so that the resistance is actually removed. So every time that you have a resistant mutation, unless you had it at both sites at the same time, you would remove it from the population and replace it with a gene drive. So the real problem is if you were to have two mutations that happen to occur at exactly the same time. And this is something we think would be incredibly rare, perhaps so rare that it might not even happen on the entire timescale of a gene drive release in Africa. And those two mutations would have to work with each other, which again might be even less likely. So I can tell you that we've actually managed to create this multiplex gene drive. And when we've tested it in the laboratory, it works incredibly well. It actually copies more efficiently than the original gene drive we made. And those mosquitoes seem to be faster at spreading. And actually they show improved fitness, which was something we didn't expect. These are unpublished results, but something that we hope to get out in the next year or two. So you might think, what next? So in order to go from a gene drive that works in a laboratory setting to something that we could release in Africa, we need to go through very, very careful testing, and it needs to be a stepwise, phased approach. So the first thing is laboratory studies. But after laboratory studies, we need to move to large cages. And the reason is mosquitoes in a small cage do not reproduce all the normal biology. They don't need to search for food. They don't need to find a place to hide from the sun during the day. They don't need to find mating swarms. They don't even form mating swarms. So what if the mutations that we cause are actually disrupting behaviors that we're unable to measure in a small cage setting? So part of the process to getting towards a final product is actually to test these in a much larger scale, still contained, still in a laboratory setting, but such a large setting that mosquitoes reproduce natural behaviors. After that, we would move to small-scale releases in Africa, potentially enclosed within a cage setting again. And finally, we would go to large-scale releases and then surveil these after release for the months and years that it takes to have an effect upon malaria transmission. So we've built a number of these large cages at a facility in Italy, in Terni, an institute called Polo GGB. And these cages, as you can see in this picture, they're very large. They're actually enough that you can walk around inside them. And in each cage, we have enough area that the mosquitoes form these natural behaviors like swarming, and they show resting behavior, and a number of complicated behaviors that in the past, mosquitoes that have been genetically modified do not exhibit these complicated behaviors very well when you test them in a large cage. So we released our double-sex gene drive in these large cages, and we tracked them and looked to see, does it work in this more intensive testing? And we found that, again, on the bottom, you'll see the spread of the gene drive, and on the top, you see the number of eggs produced by that cage. So initially in the control, we were having around 10,000 to 20,000 eggs being produced every generation. And we saw the same thing after the gene drive release, but over time, the gene drive spread, it reached fixation, and egg production plummeted from tens of thousands down to zero. So we've now shown that the technology works not just in a small cage setting, but it works in a setting that is designed to reproduce the conditions of Africa as closely as we can in a laboratory. These results are being prepared for publication right now, and it's something that we're very excited about because this is one of the final steps before you think about moving beyond laboratory testing in a laboratory in Europe, and you think about how do we move the technology into Africa. Now, before I go into how we're progressing towards testing in Africa, I'd like to quickly highlight a recent result that we've had that we've just published in Nature Biotechnology in the last month. And it's a different type of gene drive. It's called a sex-distorting gene drive. And it works on a principle that you can bias the sex ratio of mosquitoes so that instead of sterilizing females, you can make it so that males produce only males. And those males would produce only males, and those males would produce only males. Eventually, you'd have no females in the population, and you'd have a population crash. So it works as follows. During meiosis, you have the production of sperm. And you start off with a single diploid cell, and that cell splits up into two, one which will contain the Y chromosome, and one which will contain the X chromosome. So it's possible for you to express CRISPR or another endonuclease which can cut and destroy the X chromosome without affecting anything else. And if you program that at the right time, you can destroy the X sperm without affecting the Y-bearing sperm. And what that will result in is a bias towards males. Because if you don't have any of these X sperm left, all of the sperm that will fertilize the eggs will be Y sperms, and therefore you'll only have male progeny. So the way we were able to do this is we programmed these two different enzymes. One was based on CRISPR and another one was called IPPO1. And we programmed them to recognize a sequence which is present on the X chromosome, but it's not present anywhere else. But normally when you cut a chromosome, you don't expect that chromosome to be destroyed. So the reason why the X chromosome is completely destroyed is because the sequence we cut is present more than 400 times on that chromosome. So it shreds it into pieces, so much so that it's unable to repair. And all of the sperm that contain that destroyed, shredded X chromosome, they die. And the Y sperm are the only ones that are allowed to survive and fertilize the eggs. So we took this technology and we combined it with the gene drive that I've been presenting earlier, the gene drive targeting that double-sex locus. And the idea is that instead of having a gene drive where half the progeny are female, half are male, and it spreads throughout the cage, and then eventually causes females to be sterile, we would have a situation where it produces almost entirely males. And so as it spreads, rather than producing sterile females, you produce no females at all. Now, it seems like it's causing the same outcome. It's causing a suppression. You have a reduction in the number of mosquitoes. So in that way, it's exactly the same as the previous gene drive. But what makes it different is that a female can still transmit malaria even when it's sterile. Whereas with this technology, with a sex-distorting gene drive, you don't have females in the first place. You only have males. And so you have no transmission of malaria during that crucial period while it's spreading. So even if you don't have a huge reproductive load on the population, even if you don't suppress it to such a high level, you would still reduce the amount of malaria because there would be fewer females and you would have far more males. We were able to build this gene drive and we tested it in a cage setting, in a small cage setting. And again, we saw that it spread very effectively. So we released it at 10% frequency. This is the lowest we've ever tried releasing a gene drive, and it spread incredibly quickly. And as you see in the middle panel, the number of females went from 50% down to 0% as gradually they were all turned into males. And obviously, as they were turned into males, they were unable to reproduce, and so the population was destroyed and the number of eggs went from tens of thousands down to 0. So I want to present this last technical slide, and I think it's one of the most interesting ones of the presentation and a real highlight to finish on. So what do you think might happen if we were to take these mosquitoes and release them in Africa? Now, what we have is we have a section of Africa that includes the country Burkina Faso. And on the right, we have Burkina Faso outlined in red, and every single one of these dots represents a population of mosquitoes. And so this modeling simulates what would happen if we were to release our gene drive, one that distorts the sex ratio. It's not exactly the same as the one we created, but it's a similar principle. And if we were to release it once, what would happen over time? How fast does it spread? How quickly does it suppress the population? And does it eradicate all of the mosquitoes, or do we just get suppression? Or does it fail? So each dot is a single population of mosquitoes across the entire country of Burkina Faso and the surrounding region. In red, this means that they are unmodified. In blue, it means they are only gene drive. In green, it means it's a mixed population of gene drive and wild type. And grey would mean that population is completely extinct. We've eliminated it using the gene drive. So take a look at this animation. What we see is a 10-year period. And after the first 2 years, you see that it goes very, very quickly from red, where they're all wild type, to green, where they're mixed, to blue and grey. And that means that most of the mosquitoes are either carrying the gene drive or that population has been eliminated completely. But what's really interesting is that we have not eradicated the mosquito. Even after 10 years, you see it bouncing up and down. You see that the population is never eliminated, but you eliminate small populations across Burkina Faso and across Africa, but you do not eliminate all of the populations at any one time. And it's very important to get this across. But what impact might it have on malaria? So actually, this graph at the bottom it shows that the number of mosquitoes is going from 5 times 10 to the 9, that's 5 billion mosquitoes, down to 5 million mosquitoes. That would be a 99.9% suppression in the total number of mosquitoes across this region in Africa from a single release. We think that would be enough to eradicate malaria in Burkina Faso from one release of gene drives. Of course, in reality, we would want to do the best thing we can, and we may want to release large numbers of mosquitoes and in many, many regions across Africa to give it the best chance of working. And there are many unknowns. There are many things that we do not know. We do not know if there are other forms of resistance which may exist, but this is something which is potentially a best case scenario. So how do we get to release? So we need to work with local, national, international guidelines and regulations. We need to do phase testing, and that always needs to start in the laboratory. We need to use models to inform us. We need to use stepwise field testing. So when we go to Africa, we will start with non-drive mosquitoes, something that will not spread, purely so we can test those genetic components without the risk of it spreading in a way that we can't control. We're working with communities and stakeholders. We have an ethics advisory committee, and this has been going on for almost a decade before we had a gene drive technology ready. We're actually working with scientists across Africa and outside of Africa, building capacity. We're building the next generation of scientists, and we're generating new skills in a very collaborative way. And I think this is exactly what we should all be doing when we think about our research. And that when we go towards getting technology from the lab to the field, there needs to be rigorous safety testing, independent risk assessment, and we need to address the ecological concerns, such as what would be the impact of eliminating large numbers of one species. And we've made huge progress already. We've already imported the first transgenic animal into the whole of Africa, and we've performed releases of these mosquitoes in Burkina Faso. They're not gene-drive mosquitoes, they are entirely sterile, but it allows us to show that we can rear these mosquitoes, we can track them over time, and we can do the testing that sets the scene ready for a gene-drive release, perhaps in the next decade or two. So I hope I've convinced you that gene drives can be an effective tool for malaria control, and they're complementary. It's not one or the other. We don't think that gene drives are a silver bullet. We think they could complement existing interventions. And gene drives are potentially very fast acting, they're self-sustaining, and they're very, very cheap. But another thing is that right now, one of the main problems with malaria control is that we cannot reach those who need it the most. There are many people who are in areas where there is conflict going on, or perhaps they're in areas which are difficult to access. The gene drive is spread by mosquitoes. They will spread into all of the regions where you find the malaria mosquito, whether we're able to access those regions easily or not. So this is a technology which would help everyone. And whilst the use of insecticides can be destructive to the ecosystem, we think that gene drives might actually help to reduce our reliance on things like insecticides that are broadly targeting a number of different insect species. And we can actually target just those malaria mosquitoes that are dangerous without affecting the non-dangerous mosquitoes and the other insects which are not causing disease transmission. So with that, I'd like to thank many people, including the funders, primarily the Gates Foundation and the Open Philanthropy Project, DARPA, and the Wellcome Trust, who now fund my future research, as well as members of the Crisanti Lab at Imperial College London, collaborators at Imperial, Polo GGB, Oxford, Strasbourg, Cambridge, Oxford, and the whole of the Target Malaria Consortium, which is more than 180 people, about half of whom are actually researchers working in Africa. And a special thanks to these people who've had a particularly important part in my personal development. We've got Andrea Crisanti, Austin Burt, Tony Nolan, and Nikolai Wimbeckler, who were my mentors. And with that, I'd like to open it up to questions.
Thank you very much for a fantastic talk, Drew. Obviously, we lost you a little bit at the beginning, and I wondered if maybe you wanted to summarize what we think we missed, which was basically what you're showing down there, the generation of the red mosquitoes, so the bit between the introduction and the beginning of your information about the molecular biology of the gene drives.
Sure.
You don't need to go back to the presentation, just give us a summary of that, essentially.
Sure. So, I mean, really, I wanted to use it as an excuse to show some fancy videos of glowing mosquitoes, because I think it's quite an exciting part of the job, you know. So we actually inject mosquito embryos, and they're about the size of a full stop or a little bit smaller. So it's a very difficult and challenging thing to do, but it's something that we're able to do, and it is possible to genetically engineer the mosquito. And what I wanted to emphasize is that whilst we can introduce genetic modifications, there has not been any technology which would allow those modifications to spread in the wild, and that was why we needed gene drive. So you can introduce modifications that can make your mosquito unable to transmit malaria. You can perhaps change their behavior so that they don't want to bite humans. But with all of these, you need to think, how would that actually get into Africa? How would that change those mosquitoes in the wild?
Okay, great, thank you. So we have a lot of questions. Quite a number of the questions are to do with something you mentioned at the end, and that's the ecology of the mosquitoes. And essentially, what would happen if you eliminate these mosquitoes by the gene drive system? Would it have any wider effect on the ecology? Have these mosquitoes got some other important function, some niche in the ecology?
So I mean, I think it's a fantastic question. It's something that I think we could dedicate a whole talk to on its own. But I think some important things to highlight are that we don't actually anticipate eradication of the mosquito. It's actually only going to cause a suppression. It would be a substantial suppression, but not complete. Another thing to consider is that there are 3,500 mosquito species, and we'd be targeting just the small handful, perhaps 3 or 4, that transmit the majority of malaria. And that those 3 or 4 mosquito species are actually almost indistinguishable from a number of other mosquito species, which to the naked eye, they look almost exactly the same. They fulfill the same niche as far as we can tell, but they are not efficient vectors of malaria. And one of the main reasons is that the parasite has evolved a very specific relationship with these particular species of mosquito, and it's unable to effectively colonize those mosquitoes that are not good vectors of malaria. And that one of the main reasons is that those mosquitoes, they thrive on humans. So the mosquito that we're targeting, it will feed almost exclusively on humans, and its population has expanded as human populations have expanded. So this is not something which is a big part of the ecosystem as far as we're aware. It's something which is very much linked with human life. So you don't find them in rivers, and you don't find them in lakes. You find them in small bodies of water that you would find next to human settlements, often in puddles that are there for a very short period of time. They don't seem to be the main food of any organism that we've ever found on Earth. They don't seem to make up the main diet or even a substantial part of diet of any organism we've found on Earth. And as far as we know, they're not efficient pollinators. They're in fact some of the worst pollinators out there. So we don't anticipate that there's an important ecological role, but it's something that you can't exclude. So as part of Target Malaria, we actually have a very, very comprehensive project, which is looking at every aspect of mosquito biology and looking at what role they might play in the ecosystem and what the impact of reducing the numbers might be. I think in any case, you need to think there are 500,000 lives being lost due to the transmission of malaria. We are already using technologies which destroy mosquito species indiscriminately. I think gene drives offer something which could have a reduced impact on the ecosystem by making it targeted just to those species which are actually causing harm.
Okay, great. There's a question from Lavina Loom. Could wild type males be less attracted to the modified infertile females? Have you seen any evidence for that in the laboratory experiments?
So that's certainly true. So we are disrupting genes that are involved in a whole range of female development. So it's perfectly plausible that we've impacted upon a complex behavior that makes them less attractive, and we haven't detected that. So that was part of the reason why we go from small cages to large cages, and that in these large cages we are better able to reveal those sorts of attractive elements of mosquito biology. I don't think you can ever answer that in a laboratory setting completely. So at the end of the day, this is something that will have to be seen when you get to a release in a semi-field or field setting. But I think that one thing to consider is that the gene drive is actually just, a few thousand bases of DNA, and it will copy itself into the natural genome that is existing in nature. So that gene drive will actually find itself inside a natural mosquito after one generation. And a few generations in, the gene drive will be in an otherwise entirely wild type background. So I can't answer that fully yet, but I'd say it's promising based on what we've done so far.
Okay, there was another similar question from Louis Duomo, basically addressing whether the gene drive mosquitoes could face more competition in the environment or be targets of external factors? But I guess you've more or less answered that.
Yeah, I mean, I think there's something that would be interesting along those lines, which is if other mosquitoes were to take their place, if there were other mosquitoes which were to come in and become the new dominant vector of malaria. And whilst it's possible, those other mosquitoes generally do not prefer humans as a blood meal. So even if they were to be colonized better by the malaria parasite, they wouldn't make such good vectors of malaria, and we would see still a big reduction in malaria across Africa.
Okay, and there's an interesting question from Moise Khan about national boundaries in Africa. Is there some kind of treaty or understanding in Africa about releasing this sort of technology and its implementation over the continent as a whole?
So it's a difficult question to answer because this is a new technology, and so it is posing some new challenges to how we go about regulating it. I think that what we would see happening is that there would be a consensus decision reached at a African Union level, and so we're already seeing some of the structures which would allow that to happen. I wouldn't want to go about saying exactly how it's going to happen, but I think what we would want to see is a broad consensus where most African nations are in agreement that they would like to see the technology used, and that a single country would then be applying to use that technology, and we would have it first listed as a product by the World Health Organization, as something which could be used for vector control, and then individual countries would then set about applying and saying we would like to use it. They would go through their own internal regulatory procedures, and then there would be a broad consensus across African nations before any one country releases. As for respecting national boundaries, we know the mosquito will not respect national boundaries. The mosquito will move beyond any one release site. I think that's why it's important that we do reach a broad consensus before there is a release in Africa.
Okay, there's a couple of questions about the fitness of the transgenic mosquitoes. Do any of them die before they get to the reproductive stage, for example? Are they completely fit in the environment? Would you predict if they fit in the environment?
So we don't see any excessive death in these mosquitoes, but again, I don't think we've done enough to say that conclusively. But what's important is that our mosquitoes do not need to be very fit for the gene drive to spread, because a gene drive will spread even if it comes at a massive cost. So really, it needs to almost sterilize every mosquito for it to not be able to spread. So I think the idea is that right now, the fitness that we see is far above the threshold that we need for it to spread in Africa. And so if we were to test it in a more complex setting, even if there were some subtle fitness costs that we didn't measure before, hopefully they would still be fit enough to spread. That's our expectation. Okay,
and there's also a couple of questions about whether there's a risk of this technology affecting non-malarial mosquitoes. Is there any cross-fertilization?
I think it's a really very good question. So Anopheles gambia is actually part of a species complex. There are a number of species in that complex, and there is some interbreeding between them. So it's completely plausible that you would have gene flow between those different species. And so if you release a gene drive into one, we would anticipate that it could travel into the other vectors. But all of those members of that complex are vectors of malaria, every single one of them. And they're all important vectors of malaria. But there is zero cross-fertilization outside of that complex. And that's something that there is a lot of data for us to be very sure about. I don't think there's anyone in the field who thinks that mosquitoes from within the Anopheles gambia complex can mate outside of that complex. So it seems to me that it would be restricted to a small pool of mosquito species, which are the dominant vectors of malaria in Africa.
Okay, great. There's a couple of questions about the double-sex exon disruption. And one question is, does it occur in the next generation when the mosquito is already sterile?
So the mutation that would occur in double sex, when they inherit it the first time, it will actually not affect them. They will be completely normal. And it's only when they inherit that from two mosquitoes that they that they will have the full effect of a disrupted double sex. I'm not sure if that quite answers the question.
Yeah, I think it probably does. Yeah. So it's a second generation type. Yeah, it will be completely sterile. Yeah. Okay. There's a question here: have gene drivers ever been used in the wild as yet? And what are the ethics of it?
So no, they haven't been used in the wild yet, and I don't think that's going to happen in the next few years. But there are a lot, as you can imagine, a lot of ongoing discussions about the ethics of gene drive, and I think that so far the broad consensus seems to be that gene drive as a technology has incredible potential and it's incredibly useful, but that the ethics comes down to how you use a particular gene drive. So the ethics needs to be the actual product itself, the actual mosquito. So in our case, the double-sex gene drive in Anopheles gambia, rather than the ethics of gene drive more broadly. And I think that will make it easier to answer these sorts of questions and easier to make progress and to be pragmatic about it.
Okay, and again, two people have asked about your Burkina Faso simulation, and you said that you released in the simulation you're releasing the mosquitoes once. How many mosquitoes do you think will need released in the wild? Is it a practical number? And what causes these bounce backs that you saw in the profile of the 10-year model?
Yeah, so I just want to make it very clear that it was a slightly different gene drive to the one that we've actually developed, but there are many components which are very similar, and I think that the results of our gene drive would be quite similar to that. But what you actually see is that in theory just 10 males that actively reproduce in the first generation is in theory enough to initiate the spread of the gene drive according to that model. In reality, we would be looking at releasing far more than 10 mosquitoes. You wouldn't want to rely on luck. So in our test releases in Burkina Faso right now, we're able to re-release far more than 2,000, and we're aiming to have the capacity to release probably 10,000 or more at any one site. Ideally, we would have several sites and we'd be able to release 10,000 mosquitoes on several occasions, not just once. And that we think that would be far more than is needed to actually initiate the spread of gene drives in a region locally at least. And what you're seeing in the model when it goes up and down, some of this is seasonal effects, and so the malaria mosquito does change in its number very drastically through wet and dry seasons, and that's partly what you're seeing reflected in the model. And the other part is that you see that once a population gets completely eliminated, you can have reinvasion by wild mosquitoes, and they start to reproduce and repopulate. And initially, if the population is very low and fragmented, the gene drive is unable to jump back in. And it's only once that population gets large enough that the gene drive is able to jump back in and invade it successfully. So you tend to see populations get suppressed, and then they slowly start to recover. And once they reach a threshold, the gene drive jumps back and suppresses it again. I think that's why you see these bouncing up and down.
Okay, great. There are several other questions, but I think we're more or less running out of time. So fascinating questions, fascinating talk, and I'd like to thank you very much, Drew, for that talk, and congratulations on your award. But just before we finish, I'd like to mention that the Biochemical Society welcomes suggestions for future topics and speakers to feature in our Biochemistry Focus webinar series. So if you have an idea for a webinar, we invite you to submit a proposal for an upcoming session, and you can do that by looking at our website, and you can find out more information about the webinars on that, and you can propose any potential webinars in the future. So thank you very much, everybody, for attending. I hope you enjoyed it as much as I did, and good luck, Drew, with your future research. I'm sure it'll be very profitable. Thank you.
Thank you very much, and thank you to everyone who's attended.